Hi girls, this is part 2 of Mendelian genetics. We're going to be looking at 2 trait crosses, so looking at 2 genes. So we're going to pick up with our guinea pigs. Okay, we're going to cross a black short-haired guinea pig, okay, 2 genes, with a white long-haired one. Okay, so we're focusing on hair color and hair length in guinea pigs. So the first thing that you want to do when you're solving a problem, as I said before, is we want to make a nice little key. So the key we want to make, we want to kind of keep track of all of this. So we have black, which is the dominant. Okay, we're going to use a capital letter. We have white, and we have short hair. Okay, this is also the dominant. Okay, and I have long hair. Okay, and that is going to be the recessive. So we are going to create our genotype so we know what we're crossing. Okay, this is just taking the combination of alleles. We need to decide if we're working with homozygous or heterozygous. So I know that I have a homozygous black. Okay, so homozygous black, short-haired, so it should be, it isn't specified, so we're going to assume it's homozygous. Okay, and then we're crossing it with a white long one. Okay, in order to be recessive, it has to have both alleles and lack the dominant. B, little s, little s. Okay, so these are going to be our parental generation. Remember, the first thing we have to do is we have to figure out what the gametes are. So we can set up our Punnett square. That's basically what the Punnett square is doing. Remember, each parent can give one allele, not both. Okay, because both of the alleles are the same, as long as I have a B and an S in each gamete, I'm good to go. Okay, so this parent is going to give a B or a capital B, and a capital S or a capital S. So it's going to be BS for both. Kind of an odd combination together. And then this one's going to be little b, little s. So if I set up my Punnett square, remember that this is just showing all the possible offspring, because I'm taking, if I just make this one my female, and my male, and the egg, be the sperm, egg, and so with the sperm, you get your zygote, and that's basically what a Punnett square is showing us, possible offspring based on different gamete combinations. So this will be my F1 generation, okay, and in my F1 they are all going to be, okay, the dominant, so they're all going to be black and have short hair. I wanted to write this as a ratio. Write 1 black short to 0 white long. And we could do other things. I could do like 100% black short, or all different types of things, or fractions. Fractions part, that's best for this one. Okay, so if I want to figure out my F2 generation, this is where it gets a little bit tricky. Okay, for my F2 generation, I'm going to take and cross 2 F1s together. So I'm going to cross 2 of these. So remember, this is just showing the possible offspring combinations, but these individuals might actually have 10 little baby guinea pigs. So maybe I'm going to cross one of the females, one of the males. They all are genetically the same, but there may be more than one. Okay, this isn't showing the number of offspring. It's just showing the genes of the offspring, the possibilities. Okay, so, oops. I'm going to cross, okay, one F1 with another one, ESS. And note how these individuals are both heterozygous, okay? 2 different alleles for 2 traits. We actually call this a dihybrid cross, okay, because I have hybrids, also another name for heterozygous. So I have 2 heterozygotes that I'm crossing. And when we get to the end, I want you to kind of watch what ratio I'm going to get. The ratio that I get at the end is going to be something you're going to see every time you do a dihybrid cross. So if you see a dihybrid cross later, chances are you don't actually have to even do the math, okay, or draw the Punnett square or do all of this, because you're going to be so comfortable that you can just recognize it right away and not even have to do all the crossing. So we need to figure out what our gametes are again. Okay, so parents are both the same, so I'm just going to show this two different ways to do this. So BBSS, you can use the foiling method to figure this out. And basically, you want to make sure every gamete should have a B and every gamete should have an S. Okay, you want to account for the color gene and you want to account for the hair length gene. If you lose one of them and you just have B's inside your final gamete, you're not doing it right, and you're not accounting for what's actually happening during meiosis. So I'll show you what this looks like in meiosis in a second. Okay, so to foil, we want to make sure we have a B and an S. Okay, you could have the big B, oops, and a big S, or you could have a big B and a little S. You could have a little B and a big S, or a little b and a little s. So your combinations would be big B, big S, big B, little s, little b, oops, big S, little b, little s. Okay, I have 4 combinations. And you're probably looking and thinking like, well, I didn't even think I could get 4 combinations out of that. I'm going to show you with meiosis how this would happen. So this is my starting cell at the beginning of meiosis. Okay. Okay. And this individual is heterozygous, so they have one B allele, one little b allele, okay, on a homologous pair. On a different chromosome, remember I said these are on different chromosomes, I'm going to have a big S, and I'm going to have a little s. Okay, remember that during meiosis, we will do some replication. Okay, and then during anaphase 1 of meiosis, we're going to separate these. Okay, giving rise to a cell that no longer has homologous chromosomes. Note, I'm going to cancel the right. I have a chromosome with a little b. I have a chromosome with a little s. Okay, going the other way, I'm going to produce a cell with a big B and a cell with a big S. And I know S's are kind of hard with big or little. Okay, if we go through, remember meiosis 2, we're going to separate these so then we don't have a replicated copy. But essentially, you're going to get a BS and you're going to get a BS. But remember, okay, independent assortment is going to matter. The independent assortment is how those chromosomes line up on the metaphase plate. I drew them one way. They could possibly line up a little bit differently. And I'm just going to have them replicated just so, save myself a little time, so I could have a big B, little b, little b, and then maybe these two flip-flop. So this is going to be a little s, and this one's going to be a big S. Okay, note how they're on different sides. So if we go through anaphase 1, the resulting cell is going to have little b, and it's actually going to have a big S. So my final gamete would be little b, big S. Okay, and this one, well, after I go through meiosis 2, you're going to separate your replicated chromosomes in meiosis 2. Over here, I'm going to have a big B and a little s. So big B, little s. Okay, so here's my one possibility, 2, 3, 4. Okay, all different. Foiling looks a whole lot easier than drawing this out. But independent assortment, okay, how the chromosomes line up on the metaphase plate is really going to determine, okay, what kind of combinations we get. Okay, and this is just with 2. Keep in mind, the more options you have, okay, the more genes you're working with, the more possible gametes you're going to get. Okay, just a little trick to figure out the total number you should get. If you look here, okay, I have 2 different choices, so I have 2. Okay, 2 different choices, 2. Okay, so my total number of gametes should be 4. Just kind of a good way to check. So if I had something like A, B, B, C, C, okay, give big A or little a, so I have 2, big B or little b, so I have 2, big C or little c, so I have 2. So this one would actually give me 8 possible options if this was the individual I was working with. Okay, so let's actually do this problem. Okay, so I know what my gametes are. I'm going to need a much bigger Punnett square. Okay, so I have 4 options. You really just need to do the number of options you have. So if you go and you produce gametes and you have 2 different you have 1 gamete for each parent, okay, don't make yourself a giant box. Okay, just make yourself the size box you need. So I have 4 options for each parent. So if I draw out this Punnett square, I want to make sure I have 4 sides for each parent. Okay, if you do this and you have 8 options for each parent, you are actually going to have a very large box to do. In that case, sometimes it's not always helpful to actually draw a giant Punnett square, and I'll show you tricks for that later. Okay, so I'm going to put all my options, big B, big S, big B, little s, little b, big S, little b, little s. Okay, I'm just going to power through this. Okay, combine them together. Okay, so there is the completed Punnett square. Now we want to figure out what our ratio is. So you could go through and count these, but I usually like to make a key first. I'll go back and reference my key. So I'm going to do this. So anytime it has a capital B, it's going to be black. Okay, it doesn't matter what the second allele is, it'll express black. You only need 1 dominant allele. Okay, anytime there is a capital S, okay, you know that the hair is going to be short. It doesn't matter what the next allele is, so this is going to be black and short. You have a capital B, but 2 little s's. This individual is going to be black and long. Okay, so black hair, but the lack of the dominant allele is going to give you long hair. You could also have white hair. And this would be white and short, or I could have white and long. If you go through and count these, you're actually going to get a 9 to 3 to 3 to 1. Every time you do a dihybrid cross, you're always going to get this ratio. Okay, so 9 black short to 3 black long to 3 white short to 1 white long. Okay, you're always going to have 1/16 of them being the double recessive. And you're always going to have 9/16 expressing both dominant traits, and then 3/16 for each of the combinations having 1 dominant allele but having, or 1 dominant trait but having expressing the other recessive. Every single time, that's what you're going to get. Okay, so if you wanted to write out your ratio, 9 short, 3 black, long, white, short, to 1 white, long. Okay, so bring any questions you have about dihybrid crosses or Mendelian genetics with 2 traits. Have a nice day, girls.